Now then, we are going to be acting out protein synthesis today. This is a really fun, to me, activity. So it's going to involve 10 volunteers. So a good solid third of you are going to be involved. Oh, I'm not asking for you yet. I'm going to explain what you have to do first. And then you get to decide if you want to volunteer or not. The first volunteer that I'm going to need is going to be acting as our ribosome. What do ribosomes do? They make proteins. So obviously a ribosome is going to be really important in acting out protein synthesis. Now I'm also going to have a couple of other, oh I'm sorry, what are ribosomes made of? RNA. What kind? Ribosomal RNA. I know, so creative the naming. So we've got our ribosomal RNA, but there are going to be two other types of RNA that are really important for protein synthesis. What are they? tRNAs. Very good. So we're going to have tRNAs. Okay, so people are going to have to act as tRNAs. And we're going to have mRNAs, but people are not going to be acting out as mRNA. Because that mRNA transcript is simply going to be made where? In the nucleus. Yeah, it's going to be made in the nucleus. And so the ribosome, whoever ends up being the ribosome, I'm going to have them come and make the transcript over here. It will have a start codon, it will have a stop codon, and then it will have other codons in between. So we need to go ahead and make sure what your jobs are going to be. So we know that the ribosome is going to be responsible for directing the overall making of the protein. We know that the mRNA is providing us with the codons. The codons are going to be that genetic code, that secret code that is going to be necessary for determining that amino acid sequence in that final protein. So that's going to be done for us. So let's for a moment talk about the job that 9 of you will have as tRNAs. Now, as a tRNA, what is the primary responsibility going to be? What does the tRNA do? The mRNA has the message, right, from the gene. So we've got a copy of the gene that's telling us the sequence of amino acids. The ribosome is responsible for directing protein synthesis. So what do the tRNAs do?
They carry the amino acids.
Aha, so they carry the amino acids. They're the true translators, right? So they're made up of nucleic acid, but they're physically attached to an amino acid. So that's really pretty cool, because they're going to have to be the ones, they're the bridge between nucleic acids and actually making a real protein. Now, as a tRNA though, you end up as a tRNA, you're made up of RNA, and you're going to have an anticodon that's listed on your little nameplate, okay? Now that anticodon is essentially going to be like your feet, okay? Because your feet would be what sticks. If you think about the general shape of a tRNA, it looks kind of like this, right? So it's got like a feet part, that's where the anticodon is, and then it's got the kind of two little arms that kind of jut out, and actually kind of like they hug each other, and it's kind of a weird shape really. But sticking out of the head portion then of a tRNA would be where the amino acid attaches. So the attachment site of the amino acid would be like way up here. And I've had students say, oh, you should get like funny little hats and have like an attachment site. And I'm like, yeah, I don't have that, sorry. So guess what? You're going to have to hold it up over your head instead. Do you think it's important that the tRNA with a particular anticodon is attached to a particular amino acid, or do you think it's just kind of a generic thing?
I think it's important. Just a little bit.
I love these like, yeah, it's like a thanks, Captain Obvious kind of question. Yeah, so yeah, of course it's going to be important. So because the anticodon, what's the anticodon bind to? It's going to base pair, right? What's it going to base pair to? The codon. So you're going to have, this would be one side, and then you would have the codon that would look like what? What would the codon be for this anticodon right here? We have AGA, so what would we be looking for? Yeah, we would need to find UCU. Of course, we want about to be like a very loud codon here. It is. All right. So yeah, so you would have AGA on the tRNA, and then you would have UCU here on the mRNA. And it's going to be the ribosome's job to make sure that they're matched up. Now, when we're matching those up then, that tRNA that has a UCU codon, you've got a genetic code in front of you. Tell me what amino acid should be attached to a tRNA that has an AGA anticodon. And are you going to use the anticodon to look it up in your genetic code or something else? You need to use the codon, right? Because the genetic code chart that you have, look at it please. Everyone look at your genetic code chart. Because the genetic code chart that we have is based off of not the tRNA anticodon, it's based off of, I lost it, off of the mRNA sequence, right? So UCU, what is the amino acid for UCU? Serine?
Serine, yeah.
Okay, so that means the person that would be wearing this particular anticodon, which is essentially their feet, would have to look at their anticodon. So when you get your nameplate, you have to look at your anticodon and you have to think, what is the complementary base sequence of my codon going to be? Okay, UCU. You then look up in your chart what amino acid that's going to be. So in this case I go, okay, so my codon is UCU and I need to have serine. Well, what I'm going to have you do when we're acting it out is you guys are going to come stand over here and notice that you don't have, right, at the beginning, you don't actually have your amino acid. So you're going to need to go get charged with it. We call it charging the tRNAs. So at a particular point, I'll have the ribosome say, tRNAs charge. And so the tRNAs, you guys are going to all line up, you'll be right over here, and you're going to have to run over here, where I have all the amino acids displayed for you. So we've got yellow amino acids. These are going to be hydrophobic amino acids. They're going to have hydrophobic side chains. These are the kind of amino acids that you would want if you were embedding a protein into a membrane, for instance. The blues and the reds, these are charged amino acids. We find charged amino acids a lot of times in active sites of enzymes. And then I've got some white, which are polar. So they like to interact with water, and they are hydrophilic. But they also don't have, a lot of times, a lot of chemical reactivity in them. So, as a tRNA, when you are charging, you come over and you look at all of the amino acids, you find the one that matches your codon, your anticodon, your codon, you put it up over your head, and you yell, charged, and you run back over to stand with the rest of the charged tRNAs, and you wait. And basically that means that you would be just kind of floating around in the cytoplasm, just aimlessly. Do you think tRNAs talk to each other?
Perhaps.
No, molecules don't actually talk.
If I were to
So they're just floating around waiting to see if at any point there's the possibility that a ribosome might come across an mRNA codon specifying UCU, at which point they can go run over and go, me, I'm charged, I'm ready to go, and the feet can stick. Now, realizing that that is the job of a tRNA, realizing that as a ribosome, you're responsible for orchestrating all of this, I would like to see who wants to be the ribosome? You want to be the ribosome. All right, here you go. Here's your ribosome necklace. Congratulations. All right. Now, I'm really curious because now I need 9 tRNAs. And I have, I'm curious if this will be the one class, oh, by the way, ribosome, I need you to go ahead and you're gonna need to go ahead and get a transcript ready. Okay? So if you could please come. We'll start with here. Okay. Okay. Yeah. So we're going to start with AUG right there. Keep those hands high. I only see 1, 2, 3, 2, 3, 4, 5, 6, 7, only 7, 8.
One more.
Gonna need one more. Am I gonna have to pick a stick? Am I gonna have to pick a stick?
Shut up, Aiden.
If it's, oh, wait, do we have 9? 1, 2. Did you just volunteer or you had your hand up the whole time? Okay, I can always pick a stick. That's fine. There you go. You have your hand up? Okay. There you go. So remember when you get your nameplate, that's your anti-codon. That is your anti-codon. Okay. Let's see. Last person. Jaden D. All right. So remember, guys, you're looking up your codon. Okay. TRNAs don't pair. You don't pair with her. Your complementary sequence is going to be the codon. That's what you're going to look at. So what's the complementary sequence of AG? That's what you need to look up to find out what your Okay, now guys, for the audience, you're going to need to have those code charts out. Because one of the biggest misconceptions that happens is the tRNA, the person who's acting as a tRNA, doesn't understand that they're supposed to be using their codon, not their anticodon. And so they accidentally pick up the wrong amino acid. So audience, I need you guys to be watching for that. So you're going to have to look at their anticodon. You're going to have to look at the amino acid that they get charged with, and you need to verify that it's correct before we go over and start the protein synthesis, okay? Audience, you know what your job is? Okay, does everyone have their genetic code out to be able to do that? Okay, tRNAs, come over here. tRNAs, assemble! So I'm going to have you stand on the other side to direct this. And I want you to put your arms out like this. So that you're able to encompass two codons at a time. Okay? As one person. And you're going to have to read it out AUG and CAU. So you're reading out the codes. Okay? They're not coming. You have to read it. That's their signal. You're directing everything. So you're going to have to, once they're all charged, once they're all charged, you're going to have to, I'll tell you when. You're going to have to go to AG and CAU. When you get both of them here, they're going to have tRNAs, are you ready to charge? Yes. Okay. Okay, I need everyone quiet down. We are going to start this process now.
tRNAs, charge! Charge!
Yours, what's your code?
I'm charge.
This is UAC.
Oh my god, bro.
You didn't even write.
Oh
yeah, above your head.
Dual time. This one.
Okay. Remember to hold them over your head. Now, audience, this is where your job becomes really super important. You need to look at all of those amino acids and realize, don't cover up your anticodons, and realize that you need to think about the complementary base sequence to the anticodon that is on their feet, essentially, and does that match with the correct amino acid that they are displaying over their heads? Give you a moment to look at that. All right, now tRNAs, well, they're double checking. The ribosome is going to basically have two nice comfortable spots for charged amino acid tRNAs to come and stand. She is going to read the codon to you. So she'll read out the codon. When you hear your codon read out, then you come over with your amino acid and you stand right next to your codon, okay? So in the beginning, once there are two individuals that are standing there, then she is going to catalyze peptide bond synthesis between the two amino acids by saying peptide bond. Say peptide bond.
Peptide bond.
Very nice. So we've got peptide bond. And so the person that's standing to the right will then hand over their amino acid to the person standing on the left. I want you to put that amino acid behind. So your amino acid goes behind that growing polypeptide chain, okay? And then once they're not charged anymore, that's the ribosome signal to shift. So you will physically move over one. Just one. There, no, you moved over two. There you go. So once she's moved over one, you're free to take off your necklace, put it down, and go sit down. At that point, she is going to call out the next codon. Person needs to come, stand there, and that same process is going to happen all along the way. Okay? So, are you ready, ribosome? All right, then. Ribosome, attach.
AUG. CAU.
Oh, my bad.
Peptide bond.
So you need to direct it. So make him give that amino acid to behind this. All right? And now shift. Shift, and you are free to go. All right? Take off your necklace.
UUU. Peptide bond. CCC. Peptide bond.
That's right.
Yeah. CGG.
I have the wrong one.
You have the wrong, and no one else noticed?
I noticed and I told him.
Audience!
I told him.
So what are you supposed to have?
CG, I think.
Guys, look it up. You have
You're
one
supposed to
job,
have
audience. One job.
Arginine.
And you have what, Alanine?
Arginine.
Okay, hold on, guys. Let's get arginine. Yeah. Okay, wait. Guys, I actually would like to use this as a teaching moment, okay? Because I'd like to point out the amino acid that Rishi had is alanine. What color is this paper?
Yellow.
Okay, do you remember what that is? What that means about the side chain? It is
Hydrophobic.
hydrophobic. Do you remember what blue is?
Hydrophilic.
Charged. It is charged. Do you remember that I said that charged amino acids are often found in active sites of enzymes? This is like vocab term day, right? So the active site then, that's what catalyzes chemical reactions. So replacing something like a charged amino acid with a hydrophobic amino acid, do you think that protein is going to function the same way?
No.
No. That is the easiest way to completely ruin a protein. Make it completely non-functional. Good job, Rishi.
Let's go.
All right, keep going.
AAA. Peptide bond.
No, this
Yours goes in front.
It goes in front.
But that's okay, that's the ribosome's job to do.
ACG.
It's mine.
Peptide bond.
On the test, are we gonna have like a chart?
AUC. Peptide bond.
Now, I'll take it.
UCU.
Other way around. Read it.
QAA.
That's a stop codon. I'm gonna act as a termination signal. All right, now Terminator, I like to call him Terminator. Terminator is going to come along, come into that site, and basically disconnect the polypeptide and actually disassemble the ribosome. The whole thing basically falls apart. So, bye ribosome. Oh, she's falling apart right there. Good job. I like it acting up. And you're free to go too. So that polypeptide then, oh my gosh, if it, there are two different possibilities for that polypeptide at this point in time. It can either spontaneously, in the cytoplasm, spontaneously fold into the proper conformation. This is something that we're going to model, because in the cytoplasm, you know how there are different side chains that have different kinds of chemical natures to them? They can be polar, they can be non-polar. They can spontaneously fold into some protein that's going to be active. Or a chaperonin can come along and help to fold it in the appropriate manner. If the ribosome were attached to the rough endoplasmic reticulum, the way that would work is one of the initial sequences of amino acids would contain a signal that says go to the ER, at which point the whole ribosome and the little polypeptide that was coming out of it would basically be escorted over to the endoplasmic reticulum, where it would attach and deposit essentially the polypeptide into. That's for proteins that need to be packaged into vesicles, or they have to be packaged into membranes because they're membrane-bound proteins of some kind. And then there would be additional issues that would happen as they got processed along the way. But in essence, you guys just acted out protein synthesis. Everyone give everybody a All right. I'm really, really proud of you that to just come up and do that is good. Oh, you know what? Your job's not done. Would you mind going ahead and picking up that that mRNA transcript for me? That would be awesome. Thank you. Okay. So any questions about protein synthesis at this point in time? Yes, Jade?
Okay, so the green things on the floor, that's like like,
That's the mRNA.
yeah, so it's like, that's like the original thing, right?
Well, the original thing would be the DNA gene.
Yeah.
So it is a copy of the gene. Yeah, and so that transcript, where's the mRNA going to be made? In the nucleus, right? So we're going to have that whole thing, the DNA opens up. We haven't gotten into the nitty-gritty of transcription yet, but the DNA opens up. We have initiation, where RNA polymerase is able to come and bind. We've got elongation as we're making the transcript longer, and then we've got termination that happens. Once we've got a final, you just said it there, thank you. Once we have a final version of the mRNA, in a prokaryote, it's going to be the final version. In a eukaryote, there will be some processing that we have to discuss with exons and introns. The introns getting cut out and the exons getting stuck back together. That mRNA, then once it's completely processed, exits the nucleus and goes into the cytoplasm. That's where we started. So that mRNA transcript, that was the green stuff that was on the floor. And then we had the ribosome that came, bound to it, and then you guys were acting out as tRNAs. Seem hard? Not too bad? Okay.